Today I'm going to talk about biological networks and finish our discussion about bioinformatics this semester. So I'm going to mainly talk about how do we get these biological networks, how do we know which proteins are interacting with which other proteins in these pathways, and if we have such a genome scale network of an organism, what do we do with it? Okay, what kind of analysis we do on such a network, how can it help us? I'm going to talk about these today. So, as I mentioned yesterday, there have been low throughput studies on specific pathways, trying to find out all the members, all the proteins associated in a pathway, and only focusing on that pathway. And these results are usually published in articles, scientific articles. And there are now, when we talk about biological networks, our goal is to collect all these pathways that are published in different places or maybe in some databases as stored separately, to bring all of them together to create a more holistic view of a single organism. So, okay, we have these pathways. For example, can some proteins within this pathway be interacting in another protein in the other pathway? So when we bring all these pathways together, what we have is a very large scale network of an organism, which may show us some important things about the function of the cell. So this biological network that I showed yesterday was just a small subgraph of an actually very large graph. In this very large graph, we have a node for each protein in the organism. So imagine in that graph, there are going to be thousands of nodes. Even visualization of such a large-scale network is a problem. I mean, people have been devising different algorithms to view such large-scale networks. I mean, just imagine 10,000 nodes, and there are hundreds of thousands of edges between those nodes. Trying to visualize it on a computer screen. It will probably be something like this. I think I have that figure here. Yeah, it's going to look something like this. And even this one is a small network. It contains only 3,000 nodes and 12,000 edges. If for the human network, for example, in I think HPRD database, Human Proteome Reference Database, contains about 15,000 or 20,000 edges and 80,000 nodes. So it's much larger than this network you see here. And as you can see, visualizing this like in this layout does not give you any information about the organism. I mean, you cannot understand anything about, it doesn't look like a very well organized circuit, right? Circuitry board. It just looks like a point cloud or a dust ball. It doesn't look like anything. So visualizing, but why doesn't it mean anything if we visualize it like this? This is just a particular layout of these nodes and the edges between them. The underlying data, the graph, and the way you visualize it are two different things. Okay, I can view the same graph. A graph is a tuple of vertices, a set of vertices, and a set of edges. Okay, a vertex has a unique label. You can identify. So these are sets. So here, for example, this one is a graph. A, B, C, D. This is my vertex set, and my edges. My edge is. There are different types of graphs. For example, edges may be directed, undirected. Edges may be weighted. You can have weights even on nodes, but usually on the nodes you can also have some kind of other additional information. But generally, when we talk about graphs, usually the main distinction is whether you have a directed graph or undirected graph. Directed graphs are graphs in which edges have a certain direction. From a certain node to a certain node we have edges. In undirected graphs, the edge does not have a direction. We just have a direction between these two nodes. And for example, protein interaction networks, the edges are usually undirected because the semantic meaning of interaction, this protein interacts with this protein, is undirected. Okay? I mean, these two interact with each other. And when you say these two interact, you don't give any direction information in this sentence. Therefore, usually protein interaction networks are undirected. If I have an undirected graph, if I put my edges here, the edge AB and BA are the same thing in an undirected graph. So I have an edge between A and B. This is not an ordered tuple. What I mean is, so I write my edges like this. Okay, so this is my graph. A graph, these sets, the set of vertices and set of edges has the complete information about this graph. Okay, now I did not provide you any visualization of this graph. And the visualization is an independent part of a graph. I mean, it's how you visualize it. Okay, I'm going to have nodes like this. A, B, C, D. I'm deciding on such a layout is just an option. It's a layout and it's independent of the underlying graph data. I could have visualized the same set of nodes like this as well. A, B, C, D. Okay, where you are going to put these nodes on a 2D plane, on a 2D image, is about the layout of the graph. There are different layout algorithms which tries to capture the underlying relationships between nodes. For example, in this layout, if you put your nodes like this, the next thing is you need to show your edges in between them. You can show your edges like this, or you can even display your edges like this as curved arcs or direct lines. So it's just about a particular way of visualization. So we have an edge between AB, we have an edge between CD, we have an edge between AC, and we have an edge between DB. So in this layout, the graph looks like this. And let's see how it's going to look like in this layout. We have an edge between AB, we have an edge between CD, we have an edge between AC, and we have an edge between DB. Let me do it like this. Okay? So these two graphs are the exact same graphs. Okay? If I show you this one and if I show you this one, they are exactly the same graphs. I'm just, by the looks of it, by looking at a 2D image, you cannot say that they are different graphs. They are same graphs, and there is a difficult problem in computer science, which is the graph isomorphism problem, is if I give you two graphs and I do not give, in these two graphs the labels are different. I use a different label set for the second graph, for example. Let me just, for example, use X, Y, Z, W. Okay? If I change the labels. Now the question is, are these two graphs still equal? Can you find me a correspondence between their labels? But I mean, the problem is, can you find me a correspondence between labels which is going to make these two graphs equal? Saying that, for example, if you make A is equal to W, B is equal to Y, C is equal to Z, and D is equal to X, if you make this correspondence, the graphs will be equal. Such graphs with different labels, but with equal topology, they're exactly equal, are called isomorphic graphs. Okay, they're isomorphic, and identifying whether two graphs are isomorphic is a difficult problem. And I'm not sure, I mean, subgraph isomorphism has not been shown to be, there is also subgraph isomorphism, meaning that whether a large graph, a small graph is contained exactly in a large graph with these matchings. So these are difficult problems in computer science. So the reason I told you is that, I mean, the layout is something independent of the actual underlying graph we have. This is, when I talk about graph, you should think about something like this: a set of nodes and a set of edges. And in biological networks, in the networks that we use in our research, for example, this set is usually of size thousands, 10,000, tens of thousands. For example, 10K, or typically a yeast network is close to 20K edge. So a human network, for example, has something similar to 20,000 nodes, and the number of edges is somewhere around 100,000. Okay, so just imagine these sets containing thousands of nodes, and the edgeless containing hundreds of thousands of edges. Now, if I give you such a thing, such a graph, providing a good layout for it, I mean, those 10,000 dots are not going to fit in your screen. I mean, they're not, and finding a good layout to show the relationships between them is going to be really difficult. In this particular layout that you see here, can you guess what kind of a rule has been used in this layout? I mean, it's difficult to guess actually, but does anybody want to have a wild guess? I mean, what kind of an algorithm has been used? It's not a random layout, okay? As you can see, there's some kind of a structure that you can see in this visualized graph. For example, what is the thing you see? Can you see some kind of a structure here, or does it look like random to you? No, it's not. It doesn't use k-means clustering, because to use k-means clustering, we need to have some kind of a distance between two nodes. Here we don't have that distance. We can have a distance. We can define a distance on a graph. For example, the length of the shortest path between two nodes in the graph can be used as a distance. It can be used. I mean, it's not a very bad guess. It's a good guess actually. Maybe close by nodes are put together here. But that's not the layout for this graph. It may be used for graph layout. Yes. One. No, all the graph is shown. I mean, you cannot decide on a layout. If you are going to visualize a graph, if I tell you to show me all the graph, you cannot decide to hide some of the edges. Okay, all the edges and all the nodes are visualized here. I mean, when I ask you, for example, show me this graph without any additional options, you need to show me all the edges and all the nodes. And this is what is done. All the 12,000 edges. I mean, if what you said was true, this should have 3,112 node edges here. I mean, we are seeing here 12,000 edges and 3,000 nodes. So on the average, each node has 4 outgoing edges on the average. Yes?
Some nodes are participating in many connections, and some nodes are sparse. I mean, the number of connections between some nodes and other nodes are less, but some nodes are acting as a hub node.
Okay.
This has many connections to other
nodes. Okay, so how do we use this information in this layout?
This network is called scale-free network.
Yes, but I'm talking about this layout. How does this layout use that information? So you're in the right track, but for example, where am I going to put a node in this graph? So can you see a certain structure in this layout? I mean, it's difficult to see because it's very crowded, but there is a certain structure actually. But you're in the right track.
We put the sparse
edges into the center bars and put the less edges, which have less edges, put the outer bars.
Okay, so sparse means less. Okay, so it's okay. So that's how this layout is done. So high degree nodes, meaning that a degree means the number of connections a node has. So if a node has high number of edges, high number of connections, they are put into the center of this graph. And if it has less number of edges, 1 or 2 edges, it is put at the peripheral region, at the boundary of the graph. So this is that kind of visualization. I mean, some nodes in the, you cannot see it very clearly because of the projector, but there are some nodes in the center, and we cannot, I mean, if you look at the edges around them, it's just all gray. I mean, we cannot distinguish individual edges. They have a lot of edges around. So it's, in this layout, what they do is they put nodes with many connections into the centers and nodes with few connections around at the peripherals. Okay, this is just, I mean, you may question whether this layout is a good layout or not. I mean, for maybe certain purposes, for certain applications, I'm not only biological. Think about when we talk about graphs. Graphs appear everywhere in science. Okay, social networks, communication networks in computer engineering. So we have graphs. When we talk about relationships between two things, if we combine the relationships between many things, we get ourselves a graph. Okay, so we have social networks, we have communication networks, we have, for example, food chains, food networks, who is ecological networks, for example. So anything you can think about a relationship between two things, you can have a graph representation of it. And a certain layout may provide you useful information for different applications. There are different layouts. There are layouts, spring layout, energy-based layout. There are many different layouts existing in some visualization programs. Okay, now the reason I told you that about layouts was the underlying data and layout are two different things. Okay, and here in this picture, I think it uses spring layouts or something like that. It's using a different layout algorithm, but here we see multiple edges, for example, between nodes. So in this graph, the authors decided to, for example, there may be different evidence that indicate two proteins associated, interacting with each other. For example, evidence from microarray data. If in the microarray data, these two proteins show similar expression behavior, it may be an evidence that they are associated in the same pathway. So, for example, for microarray evidence, they use a certain color edge. For literature evidence, if in the literature there's a paper that says that these two proteins are interacting, they are also putting an edge between them in a different color. So the authors that developed this graph, which is the string data from the string database, decided to use different colors for different types of edges, for different types of evidence. So if we have many different color edges between two nodes, it shows there are a lot of evidence that shows these are interacting. And this, as I said, this is just a subgraph in a very large graph which contains tens of thousands of nodes. All human proteins are existing actually in this genome scale protein association network, and we can query this network saying that show me the neighborhood or a local neighborhood of a specific gene, and you can get that easily. Okay. And at this point, maybe I may talk about some practical implementation details. Last year, I asked this, I guess, as an exam question. For example, how do we get this subgraph, which contains the local neighborhood, the first neighbors of this gene BRCA1? Okay, so we are interested in this BRCA gene. And we look at, we have the whole genome scale network, the whole picture of the protein interactions. And what we want to do is, maybe let me make the question a little bit more difficult. Get me the nodes which are interacting with BRCA1, get me the proteins, and also do this in, for neighbors of in a distance of 2 edges. I'm meaning that I also want to see the neighbors of neighbors. So give me a subnetwork in this huge network. The problem is can be associated like this, can be formulated like this. Given, so this GVE is my whole genome scale network which contains 20,000 nodes and 100,000 edges. Find me a subgraph G prime which is going to be V prime, E prime. Find me a subgraph here, G prime, V prime, this G prime, in which this BRCA1 is going to be an element of this V prime, okay? And also what I want to do is I want to have the direct connections, directly interacting proteins with BRCA1 also be member of this V prime, and also the indirect neighbors, the second, the neighbors that are 2 edges away should also be members of this V prime, okay? And after you find the first step is to find the V prime, and after you have V prime, this E prime should contain all the edges between the nodes in V prime that are that exists in this E. In other words, it's the rule to put an edge in E prime is this: if A and B are elements of V prime, okay, if and AB or BA is an element of E. Let me erase this part. Then AB or BA should be element of E prime. Okay? So this, what I've written here is that if you have 2 nodes that you find that are going to be in V prime, and if these 2 nodes are interacting in the large graph, you should put that edge in the E prime as well. So it's what you are doing is, in this large graph, this is actually how it is done. This subgraph you see here, it is what it shows. It shows the interactions between these subsets of nodes. Okay, so it's a subgraph of the large graph G. So how can we find this? And actually, it's not very difficult. I have already written the rule here. And in order to talk about real implementation of this in a programming language, we should talk about how you are going to represent a graph. What kind of a data structure can you use to represent a graph in a program? Any ideas? So, I mean, you can just have these, you can represent them as sets. Okay, you can have a set of nodes. You can have a set data structure in your set data structure, or you can have them as a vector you may have, or a list. Just have a list of nodes and have a list of 2 tuples like this. And that's one representation. But can you think of other representations? I mean, is this representation efficient? What does efficient mean, you may ask? For example, if in this representation, if I ask you, find the neighbors of A, how would you do it in this list representation? You should go over all the edges in this E, right? You should go over all of them to see whether this, and find all the tuples that contain A. Okay, this is how it would be done. I mean, if you had this as a list of edges like this, unordered, no structure at all, what you need to do is you need to go over all 100,000 edges to find the first neighbors of node A. Okay, you need to do 100. In other words, this E is when you have n nodes in a graph, the edges can at most be n square. You may have n square edges in a graph of n nodes. In other words, to find the first neighbors of A, you should have an algorithm that requires O(n) square time. So this representation, therefore, I think is not efficient because I should be able to find it really quickly. If for a node, if I'm asking for a node, which are the first neighbors of this node, I should be able to find it at an instant time, in O(n) time. How can you reduce this O(n) squared to O(n), and what kind of a data structure can you use to represent a graph? Okay, if we have a linked list, what would the linked list contain?
It will contain both all the nodes.
Mm-hmm.
It will contain all the proofs from one node to another. So
Okay. Okay, you can. What you mean is that you can have the graph. So have the graph actually represented as with links as exactly. I mean, with no intermediate abstraction, we can have a node as a structure or a node class, for example, and we have another node which is interacting with this node, we are going to have a link to this other node. But the number of links that leaves a node is variable. How are you going to do that? I mean, if it's in a linked list, if we use the linked list structure, we usually in a linked list, if it's a singular linked list, we only have the next node. Or if it's a double linked list, we also have a previous. We have a next or previous in a linked list, but here we may have multiple, as you can see here, a node may have multiple connections, multiple links to other nodes, and this number is variable. It's like, it also looks like a tree with a variable branching length, okay? But it's not a tree because we can have links back to the node itself. In a tree, there's a root, and you cannot. There's this strict parent-child relationship in a tree. But we can have, for example, this one to be a parent of this one, which is not allowed in a tree. Such kind of a link will not be allowed in a tree, but we can have in this graph. Yes? Okay, that's a very good idea. So for each node, we can have a variable list which only shows the neighbors. So we can do it during construction. By just one pass on this graph, we can build a data structure to ourselves, so later queries would be easy. So that's a good idea. And I'm going to show you a representation which is very convenient to represent a graph. I mean, a graph, the whole graph actually can be represented as a matrix. Okay, you don't need to consider difficult data structures. You can represent the whole graph as an n square matrix, m by n matrix. If you have n nodes, it can be an n square matrix, and a cell in this matrix shows whether those, the cell ij shows that whether protein I and protein J interacts. You may have a matrix of zeros and ones. If they don't have an edge, you have a 0 there. If you have an edge, you have 1. Okay, so this is called, such kind of a matrix is called adjacency matrix. Okay, so the whole graph can be represented, and this adjacency matrix representation is very efficient, and we can do many things on this matrix I'm going to show. So in adjacency matrix representation, you have n nodes here, and an entry ij shows whether protein I, if we have a, actually I can have it like this. Let me name this matrix A. A ij is equal to 0 if ij is not element of E. It's equal to 1 if ij is an element of this E. So you can have zeros or ones in this matrix. It's a binary matrix, and it's also capable of showing directed and undirected graphs, both. If it's a symmetric matrix, you can see that it's an undirected graph. An undirected graph is going to have a 1 in both in Aij and Abi. Okay, so it's going to be symmetric. And if you allow, for example, a protein, a node, to interact with itself, these are called self loops. If a protein can interact with itself, you may ask, what does it mean biologically? Biologically, it means that in the cell, a protein does not have a single copy. In a cell, we may have multiple copies of the same protein. And the same protein different copies of the same protein may interact with the cell. For example, if two of them interact with each other, it's called a homodimer. So it's a homogeneous dimerization of two things. So the same two kinds interact with each other. So actually we can have self loops. Self loops are biologically meaningful things. I mean, a protein can interact with itself. If you look at the diagonal, if you allow ones in the diagonal, it means that you allow self loops. If the diagonal contains all zeros, it means you do not have self loops in this graph. And if it's a symmetric graph, you have an undirected graph. If it's not symmetric, it means that the direction is important. Okay. Also, such kind of an adjacency matrix representation also allows for weighted graphs. If the values in the matrix contains arbitrary values other than 0 and 1, okay, if it can contain, for example, 5.5, 3.3, 0.6, okay, if it can contain these values, it means that the edge has a weight. And biologically, what does it mean? If you say biologically, it doesn't mean anything. In biology, the interactions does not have weights. Maybe it may have some meaning biologically. For example, the strength of, if you may indicate, a weight may indicate different things. Maybe the strength of interaction, but in current biological networks that you can download that are publicly available, usually the weight means that the weight corresponds to our belief in that interaction. Usually the weights are probabilities between 0 and 1. And it's, for example, it's not like 5.5, 0.3.3, it's like 0.9, 0.5, 0.4. In string database, in the string database that you see here, the edges are weighted and the weights are probabilities. And these probabilities, as I said, corresponds to our confidence in this interaction. How confident are we? How sure are we? Because the string network uses a predicted network, meaning that we use some statistical techniques. I think string uses Bayesian approach. I'm going to talk about the Bayesian interaction approach next. How do we do that? And what we do is we compute a probability given all these evidences. Okay, the Bayesian probability computes this probability. given all these evidences about these two proteins, their microarray expression behavior, their literature information, for example, their locations on the genome, whether they're on the same chromosome and they're in close by locations on the same chromosome. This may also be some kind considered evidence. By considering all these evidences, the Bayesian approach computes the posterior probability, computes the probability that these two proteins interact. Looking at all the evidence, what is the probability that these two interact can be computed in a Bayesian approach, and it results in a probability like this, 0.9, 0.1, and it gives us, it tells us a certain confidence to our confidence value on whether these two proteins are interacting or not interacting. So we may have weighted networks like this. But in general, if you have an unweighted network, let's go back to this yeast network with 3,000 nodes and 12,000 edges. If we use the adjacency matrix representation, we are going to have a 3,000 by 3,000 matrix. Okay? And how many entries in that matrix? 3,000 by 3,000 is 9 million. Okay? So we are going to have a matrix which can store 9 million Boolean values, 0 or 1. But only 12,000 of these 9 million values are going to be 1. All the rest are going to be 0, right? I have a matrix that can store 9 million values, but only 12,000 of them are stored. So it's like 1 in 1,000. So you're wasting, you're taking 1,000 storage space, and you're using one of those 1,000 storage space. So you're wasting, 999 of the storage you got. The reason is the network is a sparse network, meaning that only a few of those 9 million possible interactions exist actually. Only 12,000 out of those 9 million. In other words, if you look at the number of nodes and number of edges, what you can see is immediately by dividing the number of edges by number of nodes, you can get the average number of neighbors, or the average degree, this is called average degree, a node has.